Having reviewed the 4 levels of protein structure and the primary structure in particular, let's now consider type of protein secondary structure. Since secondary structure relates to the ordering of backbone atoms, we want to first consider what are the options for rotating the bonds connecting backbone atoms together. Each residue has 2 bonds with free rotation, and these are referred to as Ramachandran angles. As these rotate between the various residues, it produces 2 main types of patterned secondary structure, the alpha helix and the beta pleated sheet. First of all, let's remind ourselves that there is no rotation within the peptide bond due to its partial double bond character, and these peptide bonds are represented in our figure by the green boxes. The 2 bonds with free rotation are those related to the central alpha carbon. The bond between the alpha carbon and the adjacent nitrogen is the phi angle. The psi angle is the bond between the alpha carbon and the adjacent carbonyl carbon. The first type of secondary structure that we want to consider is the alpha helix, pictured here. This arrangement allows for a linear alignment of hydrogen bonds, which you may recall makes for the strongest type of hydrogen bond. This means it produces a very stable conformation. As we review the primary sequence of a peptide, we can see that the R groups are in a trans conformation. The coil of an alpha helix is clockwise or right-handed. That is, if we place our thumb pointing toward the C terminal up, the fingers wrap themselves in the same direction as the helical coil. The linear alignment of hydrogen bonds requires that the carbonyl oxygen of one peptide bond is hydrogen bonded to the amine hydrogen of the 4th residue. This results in 3.6 residues per turn of the helix. Notice that with this arrangement, the hydrogen bonds are parallel to the helical axis and the R groups project outward from the helix. Remember, the amino acid side chains are of varying sizes and must therefore be excluded from the interior of the helix, or it could have no regular pitch. There are amino acids that influence the ability of proteins to form alpha helical structures. Recall that proline is cyclic in structure, and as such it restricts rotation. Also, since the alpha amine is covalently bonded to its own side chain, it is not available to form linear hydrogen bonds. Other factors that might disrupt an alpha helix would be strong electrostatic repulsion from adjacent side chains carrying like charges. Examples include adjacent lysine and arginine residues, or adjacent aspartate and glutamate residues. Although the side chains of amino acids within the helix do project outward, still bulky groups in close proximity can cause distortion of the helix, as in the case of the side chains of isoleucine, valine, and threonine, pictured here. There are also other types of helices that break up the regular pattern of the alpha helix. A good example of this is the 3-10 helix pictured on the right, which has 3 residues per turn and 10 atoms in the ring formed by the hydrogen bond. If we take a look down the helical axis on the bottom and compare that to a standard or canonical alpha helix on the left, we can see a very distinct structural difference. Because of the stabilizing nature of its hydrogen bonds, we find that the alpha helix is very prevalent in nature.